Hello, Sanjana. Can we start, Sanjana? Are
Hello.
you there?
Yes.
Sanjana, yes.
Good evening, everyone. Welcome to the third session of the workshop. Today, right now, we're gonna have a session, a guest lecture by Dr. Ho Liang Eng, who is currently working as an associate professor at Kansas State University. He is interested in protein crystallography, protein photonics for imaging, computational structural biology and chemistry, structure-based drug discovery for cancer and immunology. He graduated from Harvard with an AB and from UCLA with his PhD. Sir, over to you. Sir?
Hello. Hi.
Hi.
Are you ready for me to start?
Yes, sir, absolutely.
Okay, great. Thank you. Can everyone see my screen?
Yes, sir.
Okay, great. So hi, my name is Ho Liang Eng. So I'm at Kansas State University in the USA. I'll talk to you about some of our work on using artificial intelligence methods for drug discovery, especially as applied to COVID-19. So just to introduce where I'm coming from. So I'm in the state of Kansas. This is right in the middle of the United States. This is a primarily rural agricultural region in the United States. One of the something interesting is that we're also the site of the U.S. National Bioagro Defense Facility. So this would be one of the national research facility for studying zoonotic diseases such as COVID. So this center is expected to be completed by next year. So my own research is primarily in the area of what's known as structure-based drug design, especially for cancer, immunology, malaria, and now COVID-19. I'm especially interested in developing and applying machine learning methods for drug design. And our lab is a bit unusual in that we do both computational and experimental research. So here's a point already I would like to encourage. I have been working with collaborating and communicating with computational researchers around the world. And I really think it's important that computational researchers try to collaborate with experimentalists who can help them to experimentally validate their work. During this pandemic, I started a consortium called Open Source COVID-19 to do nonprofit open source drug discovery. So this is a consortium that is open to any interested scientists around the world. And right now we have more than 20 global members and partners. And we have the full range of capabilities ranging from work with DNA to chemical synthesis to biochemical assays and even viral testing in cell culture and eventually in animals. So for those of you who are working with COVID, especially those of you who are chemists and might have active compounds, we're happy to work with you for free or nominal cost to test your compounds. Again, our goal is to work as a nonprofit to develop compounds without intellectual property rights. I think we have all seen in this pandemic the great importance of not just discovering new drugs, but doing so in a way so that these drugs, as well as vaccines, are available to as many people as possible around the world. So that is our long-term goal and dream. Currently, we are focused on the two of the most well-known viral protein targets, M-Pro protease, which is involved with cutting the viral protein inside the cell and releasing it in the active form, as well as the now famous spike protein, which is on the outside of the viral capsid, responsible for engaging with the human receptor and then entering the cell. So many of you by now, of course, know that the spike protein is the target of the many vaccines that are now available and have been proven to be clinically effective. So that has been heavily studied. But at this point, there are still no antiviral drugs on the market that have been proven to be very effective. So there is remdesivir, which has been clinically approved in the United States. However, clinical tests with remdesivir show that it is at best marginally effective. So there is still a great need for antiviral drugs for SARS-CoV-2. So just want to introduce to you the very basic ideas behind AI, modern AI and machine learning. So all of most modern machine learning methods are based on what are called artificial neural networks, or sometimes just called neural networks. They are often presented in diagrams like this, which, if you're not familiar with them, can look very intimidating. But what I would like to point out to you is that it does not have to be intimidating. It is really not that hard, not that complicated. So what we have is a artificial neuron, also called a node. So what this does is that the neuron, simulates the basic mechanism of the natural neuron. The neuron has as input multiple functions. Here you see x1, x2, etc., up to xn. And what the neuron does is that it calculates a weighted sum of these functions of the input, and then it applies a simple what's known as an activation function to this weighted sum, and it outputs a final value. And so this one node can is one of these circles here in a neural network, and they are arranged in layers. So the input layer takes as input various calculations from your data set, your input data set. In the case of drug discovery, this could be, these are often very simple properties, such as some kind of molecular weight, or measures of solubility or charge, so things like that. You can also use QSAR types of features and fingerprints. All of these types of things, anything basically that you can calculate, can be fed into the neural network. So that is a big advantage of the neural network, is that you can feed into it almost anything and it can use these input and automatically calculate an output and make predictions or regressions based on as much information as you can provide, and it performs basically better as the complexity of the neural network increases and as the amount of data and training data that you provide to it increases. And so the information, the output from each layer of this neural network gets fed into the next layer, and eventually you get your output. But all this is really just this weighted sum, and then it passes through an activation function, which usually has a very simple form. So the mathematics behind it, at least at the basic level, are actually not that complicated. Now, that would represent what was shown previously would represent what's called a simple neural network, but a lot of the advances in the field have come from recent work in the last 15 years or so using much deeper, complex, large neural networks with thousands or more different nodes. And so now this is what we call deep learning that you have heard about. And so the power of deep learning comes in that if you have a lot of data, the deep learning can generally can outperform any other computational method in analyzing and classifying and performing regression on these kinds of data sets. The challenge is that as the size of your deep learning neural network increases, the computational power required is also increased, and the amount of training data that you need for good performance also tends to increase. So those two represent challenges, but as well as very active areas of research in the area of deep learning. So what is the AI actually doing? So basically, what research has shown is that these neural networks, even simple neural networks, act as a universal approximator. So basically, they can approximate nonlinear functions. And this means that one can, you can see how this works already in terms of classification. So one can come up with a function that can classify whether your data is good or bad, whether a drug might be active or inactive. And this can also be used for image analysis, because an image is basically can be represented by a function. And so as the number of neurons in your neural network increases, the representational ability of your neural network also dramatically increases. But then what happens is that you also have to be careful, and this is something that is often underappreciated, which is that as you increase the complexity and size of your neural network, there is a danger of what is called overfitting. So that means that as you have many as you have many nodes in your neural network, it can approximate any function, but it's hard to tell whether that approximation is something that's scientifically relevant. So this is basically a problem. This is the same problem as curve fitting. So here you have a very powerful multi-dimensional nonlinear curve fitting algorithm. However, if you don't have the right kind of data to feed it, then it could just draw an arbitrary curve throughout your data, and that will have no or limited predictive power, although it can give you very impressive statistics on your data set. And so ultimately, that's what you want, is predictive power. Okay, so there is my 5 to 10 minute introduction to AI and deep learning. What I really want to focus on, I'm not a computer scientist. What I am is more of a chemist, is to focus on applications of AI to drug discovery, especially from the side of chemistry. So what I've told you already is that modern neural networks can be used for classification and regression. Those are quite simple applications. So we look at what I have here as one, which is that we can use chemical structures, and often we can do, these are things we already do, such as virtual screening or QSAR, quantitative structure activity relationships. These are things that we do already. But I will tell you now that by using machine learning methods, they will, done correctly, they will always outperform traditional non-machine learning methods significantly. So anything that you can do by using traditional mathematics and multivariate algorithms can be done with machine learning and done better. What we're particularly interested in is using machine learning for another application that I haven't mentioned yet, and that is to do molecular design. So it turns out that there is a class of algorithms in machine learning that are called generative machine learning, generative algorithms. So what we're doing now is we're moving beyond the simple applications of classification and regression to actually doing design. So we're having the algorithm apply a neural network to a training set of molecules and have it learn what is a good molecule. So it's learning the rules of chemistry, and what is, and more interesting than that, what are good drug molecules. So a simple way to look at that would be that the drug molecules has properties that in chemistry we call drug-like, meaning that they are a certain molecular weight, a certain solubility, and they should be synthetically accessible. Now we can define these rules however we want, so we can add more complex rules, or we can just start with very simple rules like that are basically similar to the Lipinski's rules that you might be familiar with. So many of you probably have seen these on the internet, where there are now algorithms that can come up with entirely synthetic human faces. So all of these faces here are not real people. They're dreamed up by a computer algorithm. The computer algorithm has trained on large data sets of human faces, and it has learned what human faces essentially look like. And then upon learning that, the scientist asks the algorithm to come up with the computer's own idea of new human faces. So this represents the current state of the art, and you can see that it's very impressive. This is coming from a computer, so the computer doesn't know about the things that we know as humans, as what belongs in a face and what looks correct or natural. It just learns this from input data. If you look extremely closely, sometimes you can see graphical artifacts that can reveal that a certain image is not correct. But for the most part, for most of these images, at least a non-expert, me, for example, I can't easily tell the difference and say that one corresponds to a real person or a computer-generated face. Now, the goal is to apply these types of methods to chemistry, drug chemistry. Can we have the algorithm learn from a data set of molecules or drug molecules and have it understand what a drug molecule should look like? And then we can go back and ask the algorithm, please generate some new drug molecules. And so these new drug molecules ideally would be similar enough to the training molecules so that a scientist would not easily be able to distinguish them. We can provide more rules to the algorithm and say that perhaps please generate new drug molecules that would be similar to a certain chemical structure we are interested in. Or we could ask the algorithm to please generate new molecules that would be effective against a given structure of a protein drug target. So these are ways by which we can come up with new drug molecules for drug discovery. So the advantage of this approach is that now we are no longer limited by a priori library, in silico library of molecules for screening and calculation. This is important because the size of the size of the set of molecules, potential molecules, is massive, almost infinite. And so even nowadays, where we have the capability to screen virtual libraries of up to 1 billion molecules in size, 1 billion molecules is still an extremely small fraction of all potential molecules. So, so this gets to the point that we want to be able to sample chemical space and the diversity of chemical space in new ways. And so that is the goal and the reward for this approach. And what also helps is that as we have come up with increasingly accurate physics and chemistry based scoring functions, some of which are also based on machine learning, we can apply from simple rules of chemistry and drug likeness, or we can apply complex rules from quantum mechanics. These can all be given to the algorithm to come up with new molecules. So this is work that we're doing closely with Professor Alana Spuruguzik research group at the University of Toronto. So this gives you an example of what we mean by trying to explore chemical diversity. So again, by chemical diversity, we mean the broad combination of different ways by which atoms can arrange themselves into molecules. So because this space is massive and really cannot be explored unless one goes through the trouble and the expense and the time of actually synthesizing molecules, it really leaves us, for the most part, flying blind in chemistry. So let's say we're trying to come up with a new drug. Based on our previous experiences with drug discovery, we might think that, hey, this part of chemical space is most likely. That might represent structures that have some similarity to known active compounds. But it turns out, for example, that perhaps after this is all said and done, that the best kind of inhibitors are not anywhere near this space, but are farther away. So if we start and we're limited to exploring this space, then we will never really get to the drug that we want. So by using a generative AI strategy, the generative AI in principle has the ability to explore the chemical space not bound by an a priori in silico library. Oh. Okay. I will briefly go over our algorithm. This is a bit technical. If you're interested in the details, you can look up the paper. And in fact, there is also some code on GitHub that you can use. So this is published by the Aspuruguzik group. But what we're using here is what we call a hybrid algorithm that uses a genetic algorithm. So a genetic algorithm is used to generate molecules. A genetic algorithm is really just an algorithm for sampling and optimization. The genetic algorithm is then fed to a discriminator, which is a neural network. So this is a variant of what is known as a generative adversarial network that you might be familiar with. Generative adversarial networks are basically algorithms where you have two neural networks that compete against each other to generate a new output, a new structure. They work quite well and are among the state-of-the-art approaches for generating images, such as some of the faces you've seen. However, they are hard to train and very computationally inefficient. So Aspuruguzik group has been interested in looking at alternative ways to perform generative AI with chemistry. So this approach, the genetic algorithm, generates some molecules that are fed to the discriminator. The discriminator then judges whether that molecule is good or not based on its training on an input data set of drug-like molecules. So is this drug-like enough? Does this obey? Does this seem to obey the rules of chemistry? So one of the things that's interesting here is that the neural network here is not actually trained formally in the rules of chemistry. So because that's too complicated, we don't have a good way of teaching the algorithm how to think about chemistry. So that's performed by the neural network. After that, the discriminator provides a score as well as a separate scoring function of our own. And we combine these scores to, we call this a fitness function. So this combined score will determine how good this molecule that's generated is. If it's good enough, we will keep it. If not, it gets discarded. If it's good enough, it gets fed back to the genetic algorithm for further modification. So I will show you some of our preliminary work, and this is work in progress. So we have obviously not come up with a antiviral drug yet, but I will show you some of our preliminary work and some of the advances and challenges we have faced over the past year. So this will be in looking at drugs targeting the M-Pro protease. So we start with just a methyl group at the drug binding site of the protease. And over time, after many cycles, this molecule, you can see that it grows in size and complexity. Eventually, after 25 cycles, which is still early in the computation, you get molecules that have achieved significant size and complexity. However, if you look at these quite closely, you can see that these kinds of molecules, especially if you have some background in drug chemistry, do not really look like drug molecules. They are quite poor, actually, and they often have problems with their chemistry and represent molecules that are not realistic and cannot be synthesized. So that continues to remain a challenge for us. This is what we have been working on over the past year. We can see that since 2020 and now our current work is that we have been refining that basic algorithm to get molecules that are more drug-like. And I think we have made progress going from molecules like this, which I would say is garbage, to molecules like this, which are not that great, but are not garbage. But this, I think a drug chemist can look at this and say that, if I were to change, let's get rid of some of these phosphorus atoms, for example. And we could change here, we could change here, and now we have a molecule that is at least kind of drug-like. And these modifications are easily made by an expert chemist. So we're getting there. The other challenge that we're trying to approach is we also want to make molecules that are easily synthesizable. So that's something that we are still working on. We have made some progress, but we need to do quite a bit more. This is something that's quite hard to do, actually. It doesn't help us if we can come up with a molecule that, at least in a computer, seems to be great, but when you give it to the actual chemist in the lab, the chemist says it would take us years to produce this molecule. So that's no good. And so that represents also the point I made earlier, is to work closely with experimental chemists so that they can give you feedback on how to make your research the most useful and significant. Okay. So let's look at the results of this current molecule. We can see that it is rather long and flexible, and actually that turns out to be okay, because many of the known and most active inhibitors of the M-Pro protease are peptides and peptidomimetics, which also share this property. So here we compare the generated molecule, which is shown in maroon, with GC373, a peptide-like, one of the most active known peptide-like inhibitors. And this is from a crystal structure with GC373. And we can see that in 3D, that the AI-generated molecule shares much of the conformation of GC373. So that's good. And you can also see how well it fits into a three-dimensional representation of the protein binding surface. And when you do this, what this does is that a benefit of doing this kind of comparison is that while perhaps our AI generated molecules at this point are not yet good enough for direct use, but they can give us some ideas and clues as to how to improve the properties of GC373. So for here is an example. Look, perhaps we can cyclicize the GC373 molecule. That suggests that this would be an improvement. Perhaps we could extend GC373, again, the molecule in blue, up into this region by adding some kind of hydrophobic moiety. So already I think this can provide some kind of useful benefits to actual drug chemists. So the second case I want to share with you is not as advanced in our hands, but it's something that we're very interested in. And this is applying the same approach to peptide drugs. And I already shown you that that protease, because it's a protease and it acts on peptides, that many of the best known inhibitors are peptide-like. So in the second case, we're interested in doing the same, using this kind of approach to developing peptide-like inhibitors to the spike protein. So in many cases, peptides are important and useful for drug discovery, but often they cannot be used as the final drug compound, because peptides generally have poor what we call ADME issues. So absorption and metabolic effects. For example, they are hard to absorb orally. Peptides tend to be broken down very quickly in the body. So what we often do is that we use chemically modified forms of the peptides as a molecule that are called peptidomimetics to improve the properties of the peptides. But even peptidomimetics still often have some of the poor pharmaceutical properties associated with peptides. And so in our lab, we have been working on an unrelated project the target is the human neurotensin receptor. So this is a membrane receptor. This is what's called a G protein coupled receptor. There is a known crystal structure, not done by us, but by other researchers. It's known to bind a small peptide, and it's a potential drug target for mental illnesses as well as cancer. And so what we want to do is see if we can come up with a drug-like molecule that can replace this peptide. And so we take a similar approach as described previously, where we start with just a cyclohexyl group, and then over generations of computation, it evolves into a larger and more complex molecule. But you can see that these larger, more complex molecules already have very poor physical properties, having many benzyl groups that would lead to it being highly insoluble. And so at this point, our very preliminary result, but not very satisfying, is this kind of molecule. But we can already use this result in a useful way, although this molecule itself is not useful. When we look at the generated molecule versus the crystal structure, so here In cyan is the crystal structure, in cyan is our generated molecule, and in maroon is our crystal structure. We can see that again, the generated molecule recapitulates the conformation of the bound peptide. But the generated molecule also has some additional information that might be useful. It suggests that the binding pocket up in this region is very large, and so that one might capture more of the binding pocket by forming this kind of folded peptide. We can see that the generated molecule packs quite well in the binding pocket. And so we take the same approach, and we are trying to do this with Spike. So this is very new work, and we don't really have direct results yet to show you. So with Spike, we are focusing on this small part of Spike known as the receptor binding domain. So this sits on top of the very large viral spike protein. It's the receptor binding domain that makes contacts with the human ACE2 receptor, the angiotensin receptor. And it is this contact by which the virus is able to bind to a human cell and then enters the human cell. So if you can block and interrupt this contact, then you can block the virus from infecting human cells. So what we're looking for is a drug that binds somewhere in this interface. And so we have developed some methods in our lab for predicting drug binding sites, and they've identified a rather small binding site near residue 506 of the RBD. And here is a result from virtual screening of a small molecule that might bind in this site. However, the virtual scoring results were rather poor, so I don't expect this molecule to bind well experimentally. The challenge is to come up with a new molecule that can bind this site with higher affinity and be a better inhibitor, and that's what we're working on right now. So in general, the areas that we are continuing to focus on are to improve the drug-like properties of the molecules we are generating. We want to make sure that they are synthetically accessible, meaning that a experimental chemist can easily synthesize them in a lab for testing. One of the ideas we have to do that is in the course of building these new molecules that we can incorporate fragments from real drug molecules. So instead of just building phenyl groups and cyclohexyl groups, we can incorporate pieces from real drugs that such as indoles, for example, or imidazoles, and that could give us something that's more drug-like. Another question that we have is more broadly, can this approach be used as a test of drugability? So can we use this, for example, to ask and answer questions like the spike protein. This binding pocket of the spike protein is very small. Is it actually even possible to use a small molecule to inhibit the spike protein? And right now, our early answers is that I don't know. The scores and the molecules we have generated all look very poor. Does this mean that this is just not possible to make a small molecule inhibitor, that the only way of blocking this will be to use a large molecule like an antibody? Perhaps. So not proteins can be easily drugged. And maybe the answer is that the spike protein is just not a good target for small molecule drug design. So even achieving this answer of no is useful because it would tell us better to focus our efforts on the protease or another drug target instead of spike. But we can't rigorously say that at this point. So I want to thank the many students and postdocs in our lab at Kansas State University. Our primary experiment, especially led by Bo Wang and Bowen Tong and Louis Cabrera at our lab. Our main experimental collaborator, Troy Messick at the Worcester Institute in Philadelphia. We've benefited from the input of many former pharma scientists, such as Chris Southern and Chris Swain and Peter Kenny, who have given us a lot of advice based on their real world experience working in pharma. So here's another bit of advice from me to you. For those of you working in drug discovery, try to work with or collaborate with scientists who have real life drug discovery experience in industry. Our main collaborator on the computational side is Alana Spuruguzik at Toronto. And our funding, which comes from the United States National Science Foundation and generous support from tech companies like Intel and NVIDIA. Thank you very much for your time. I'll be happy to answer any questions.
Participants, please note, please keep your questions succinct as we have a time shortage. You can go ahead now.
Okay, sir, such a nice presentation. Am I audible?
Yes,
okay.
Thank you.
Okay, thank you, sir. But sir, actually, I want to ask one question, that how we can apply machine learning or deep learning in the molecular dynamics simulation result analysis or betterment of the molecular dynamics simulation. Although you have not covered that topic,
No.
but actually this is my query, sir. So I want to ask. So if you want to answer, so you can.
Yeah, so we have not covered that topic. That is a large area of research that we are not working on at this point, but you can look up papers by people like Frank Noe, last name Noe. He works in this field. There are a lot of scientists who are applying machine learning methods to both approximate and analyze results from molecular dynamics.
Okay, thank you, sir. Thank you so much.
Hello, sir. Hello, Abraham, one question.
Hello.
Yes, sir.
So you were mentioning about peptidomimetic, wherein you are showing the small peptide to inhibit the target protein. Is that peptide not similar to an antigen? Antigen is also a peptide. So through antigenic means, we can inhibit or we can make it to interact with the target protein. Is it not similar to that?
Yes, it is similar. Yes. I mean, the chemistry that we're using is not new. It's just the applications of the AI methods that we're using to come up with peptidomimetics and the like. It is based on traditional chemistries. Yes, sir.
Those peptides, can it be possible to be synthesized through yeast or bacteria instead of laboratory synthesis of peptides?
Yes, in principle, they can.
Thank you. It is a great lecture.
Thank you. So the peptides, so he asked whether they can be synthesized by biological hosts like bacteria and yeast. Yes, the peptides can, but if you move to peptidomimetics, then you need to do chemical synthesis.
Thank you.
Thank you, sir. Next, Kalyan.
Thank you, Abraham, and thank you, Dr. Yong. It was a great presentation. I just wanted to add about the other question one of the participants asked. We are going to cover docking and molecular dynamic simulation using machine learning that is scheduled on the 21st. So that is going to be covered.
Thank you, ma'am. Any specific questions from the participant? Yes, Harkishan, yes, you go ahead.
Sir, I have a question
Yes.
regarding the machine learning code where you have fed the data set of different molecules. Sir, what are these molecules are supposed to be, are in which format? Means in what format can we feed the molecules into the machine learning code?
So the current, that's a good question. So, I mean, the format itself doesn't really matter right now. I shouldn't say that. I mean, of course it matters. There's actually a lot of nuances to that question. Right now, the training set comes from subsets of the Zinc data set, which you might be familiar with. You can look this up online. It's publicly available. We train it using just the Zinc 2D SDF files. We have not used three-dimensional data yet, but that's something that might be someone in the future could benefit from finding ways to take advantage of three-dimensional conformational data of the training set. Another point related to this that I haven't brought up yet is the internal representation of the chemical structure. So many of you are familiar with the SMILES strings for representing chemical structure. And so what our collaborator, Spuru Guzik, has done is they developed an enhanced SMILES string format called selfies, which actually is able to better capture chemical rules of bonding and topology. And by using this superior chemical format, it helps the whole AI process because a lot of the rules of chemistry, of basic chemistry, are at least now captured in the grammar of the representation itself. So that makes for less work for the neural networks.
Thank you, sir. And one more small question, sir. Suppose I have a data set consisting of this many molecules in SMILES format. And in another column, I have this, suppose one property of the particular molecule. I want to generate a new molecule with a different property. Suppose a new value from that property. How should I proceed with that?
I mean, you can use this general approach to do that, but not out what we say out of the box using this current code. You would have to take the current code and you would have to implement a way to calculate that property. So if the current code that we're using uses the RD-Kit chemical library that you might be familiar with, and then you could, if it can be described or calculated using RD-Kit, then that you could add just a few lines of code into the current code base and do what you just described.
It means can we work directly on the structure of the molecules, or do we can we train on some property of the molecule? Can we just directly use the structure and the property?
Yeah, absolutely. Anything that can be calculated.
Okay, sir. Thank you, sir.
Just to add to this this answer, we could always use feature extraction. So if you're working on one of the structures and you want to calculate, you want to predict the same properties for a novel compound or something, you can always use feature extraction. Feature extraction methodology to it, and that's how that's one domain of machine learning that you could implement into this.
Yes, thank you, ma'am. Sonu, do you have some specific question? Sonu Benny.
Oh, yes, sir. Thank you,
Yes.
sir. So if you have to work with structure-based drug designing for a novel target, can we validate our novel structure by using artificial intelligence?
For a novel drug target?
Oh, yes, sir. If you're having scientific background for that particular target, so we have to prove it as a potential target before going for the structure-based or the target-based drug.
Right.
Yeah.
That's something we're looking at in a different project. One would have to find some way to predict what proteins make for good drug targets for a given disease, and that's not so easy. That's a more biological approach. One can use genomics data. One can use transcriptomics data to try to correlate different levels of RNA or protein with different disease states or with different drug perturbations states. So that's something we're exploring at this time. But ultimately, to validate, you would need to do some kind of experiments.
Oh, okay, sir. Thank you, sir.
Yes.
Thank you for your questions. Krupanidhi, sir, please give your concluding remarks as well.
So good evening to you all. At the outset, let me express my sincere thanks to Dr. Abraham for inviting our guest speaker today, Professor Ho-Ling Nok. Professor Ho-Ling Nok has nicely elucidated machine learning and also artificial intelligence directed drug design. Very interesting for us who are new to this field is that computationally we can generate a face of a human being after having fed the information. In a similar way, alike to that, generating a drug molecule after having provided information is a new concept. Very nicely he has generated artificial intelligence molecules, and in that he has shown very neatly GC373 having confirmations altogether as a normal molecule. It also shown something like that Q506, a peptide that is getting bound to our spike protein of coronavirus. So it's nice, sir. We want to listen and learn a sort of a computational workshop wherein artificial intelligence tools, unless we learn these things, we cannot further proceed into this particular topic. And we're all interested to join in the program of open source COVID-19.org with you all. Okay, thank you, one and all. Thank you, sir, Professor.
Thank you.
Thank you so much, Ho-Ling sir, for your great lecture. Thank you so much, sir. Okay, you can, yes. Yes, Sanjana, go ahead. Yes, now you can unmute.
Thank you, Dr. Eng. So now we have with us Dr. Janine from Mahidol University, Thailand, who will be conducting a hands-on session on how to develop a bioinformatics app. Sir?
Yes, hello.
Hi.
Hi. Okay, so shall we begin?
Yes, sir.
All right, let me first start by, may I ask the host for the permission in the Zoom app to record this workshop?
Sure, sir. I'll give.
Okay, thank you.
Yeah.
Thank you. Okay, let me share the screen. Okay, so I've shared the Notion link. And let me hide this. Okay. Okay, and so we're here. I guess everyone can see this, right? Yeah, so today we're going to be building a simple bioinformatics web application in Python. And so the example here, we're going to be using the water solubility of molecule as the topic of our web application. And so the prerequisite here is going to be basic understanding of biology and also basic proficiency of Python. And the resources that we're going to be using today is, firstly, if all of you could install Conda on your own computer. And so Conda will allow you to manage the various Python libraries, and also it will allow you to manage the environment of your Python. And I've actually made a video about how to install and use Conda here. If you click on this, if you hover on it and click on the open link, and then it will bring you up here, and then you could click on the YouTube. But then today I'm going to show you live how you could go to Conda and download it. So you want to go here, download mini Conda. Right? Let's click on it. Let's see. Let me share the other one. Okay. And now you're going to be on the mini Conda website. And so I recommend you to download version 3.8. And so a rule of thumb is whenever there's a latest version, I usually will ignore it, because when it's a new version, many of the libraries might not yet support the new version. And so if there's version 3.9, I'll just go for 3.8. And if there's version 3.8 as the latest, I would go for 3.7. Okay, so for this case, you want to go for 3.8. And depending on which version is your Windows or is your Mac, then you want to click on the corresponding link to download it. Okay, so like I'm on a Mac, so normally I would click on this one. Okay, the PKC one. It will allow you to install it in a click, click, click fashion. And if you're on a Windows 64-bit, download this one. If 32-bit, download this one. Okay. And so let's go back to the web page here. Okay. And the application that we're building today is going to be based on this particular video that I previously made on YouTube. And so you could also check that out as well. And the repo of the code that we're going to be using today will be provided here in the link column. And then click on the GitHub Repo link. So let me copy the link here. All right, so you're here. So this is the repo that we're going to be using today. So I recommend that you could go ahead and click on the green button here, which says code, and then click on download zip file. And then afterwards, you're going to unzip that. Let me share the screen. Okay, it's right here. So when you unzip it, these are the contents. And so what we have here is the Jupyter Notebook. And the Jupyter Notebook here will allow us to build the machine learning model. And then the Python file here, solubility-app.py, this is the actual web application that we're going to be building. And the Jupyter Notebook will be producing this file, pkl. which is the pickled file of the trained model. And then in order to deploy the web app, so normally the web app here, this file, will work on your local computer if you have Streamlit library installed. However, if you would like to deploy it to the web, to the internet, you need to have additional files. So you need to have the requirements.txt. Let's have a look at that. And so the requirement.txt here will essentially tell you the Python library and the version number. So here it tells us that we're using Streamlit version 0.71, and pandas we're using 1.1.3. And so the great thing of using this requirement.txt is that you're fixing the Python library version to a particular version that are known to work with your web application. So even in the future, when pandas update its version or NumPy updates its version, then it's going to work. So your web application is going to work. So you're not going to rely on the latest version. So more or less, you're kind of putting this into like a time capsule. And so if you're using this particular requirement.txt file, 1 year from now, your web application will work. Okay, so let me share the terminal. Moment. Okay, so I hope that all of you are seeing this terminal.
Yes, sir.
All right, perfect. And so I have to save this into my downloads folder, and then let's see, what's the name of the folder? Solvability. So if you're on a Mac, it will look like this. If you're on a Ubuntu or other versions of Linux, it'll look something like this. So you will change your directory to the location where you have unzipped the zip file from the GitHub repo. Okay. If you're on a Windows, you could use the PowerShell, and then you could do the same thing, cd change directory to the location that you have previously unzipped the content. And so the contents that you see here are the same that I've mentioned already. And let me open up the application. So normally I would have to activate my Conda environment. So I'll show you in just a moment how to create a new environment. But for now, we're just going to load it up. So I'll load up my Conda environment, Conda activate data professor. And then to run the app, you just type in Streamlit run, and then the name of the app. So it's Streamlit run, name of the app, and then wait for a moment, and then your application will load up in a web browser. All right. And so you can copy this link, and then let me show you. And so this thing will pop up. Okay, so the web browser of the web application will pop up. And to the left-hand side here, this is the input panel. So you put in your Smiles input notation here. So here we have 3 example molecule, right? Molecule number 1, number 2, number 3. And then this is like the logo of the web application. And then this is the title of the web app, description of the web app, and then we provided the links to the data, the paper at which it was coming from. And so here we're just gonna reiterate the input file. So molecule 1, 2, 3. And then here we're just printing out the molecular descriptor that we have computed. And we have computed the mole log P, molecular weight, number of rotatable bonds, aromatic proportion. And we compute these descriptor using the RDKit library in Python. And then it will be using a simple linear regression in order to predict the log S value. And so we have the predicted log S value here for the first molecule, this one. And then the second value is for the second molecule. And then the third value is for the third molecule. Okay, so this is the web application that we're going to be building today. And so let me show you now how to install Conda on your computer. Let me close this. Let me end this. Let me share the terminal again. Okay, so normally when it's still running, I would press on the Control-C in order to abort this. And so now let's create the Conda environment. One moment, but let me show you here. So on the GitHub repo that you see here, Data Professor, github.com/dataprofessor/solubility-app. Scroll down to the README. So this is the README file. And then you want to follow this step. So all of the instructions is provided all here. Okay. So right here, we're going to first create a Conda environment. Okay, actually, this should be solubility. Okay, and so you could copy this, right? You could just click on this button to copy it. And then we're going to type the contents here. Head over to the terminal, back to the terminal. Okay, so here we're back to the terminal. Let me deactivate my original Conda environment. Conda deactivate. Okay, now notice that whenever I am in a Conda environment, the name of the Conda environment that I am in will be in the parentheses before the $sign. And now that I have deactivated the Data Professor environment, I am now in the base environment. Okay, so I imagine that right now you have probably installed Conda in your computer. And to figure out whether you already have Conda, you just want to type in Conda. Okay, and so if you see these command here, which is like the help option, then it means that you have Conda. Okay. And so normally I just use only a few functions here. So I just use Conda followed by the name of the command that I want to do. Like for example, I want to list the package here. I could just say Conda list. And so I will see all of the libraries that I have on my computer. Okay, if I say Conda install and then like the name of a library, then it will install the library. Okay, so for now, I'm going to create a new environment. So I'm going to type in Conda create -n for new. And then the new environment, I will call it solubility. And then I will specify the version of the Python to be Python equals to 3.7.9 and then enter. Okay, and then it will ask me whether I would like to proceed to install all of these libraries. And so you want to hit on yes. Type in Y and hit on enter. All right, and now the environment has been created successfully. And so you want to activate the Conda environment by typing in Conda activate solubility as shown here. And whenever you're finished with using your environment, you could just deactivate it by typing in Conda deactivate here. So now we're going to activate it. Conda activate solubility. And so let's list the package here. And so this is a new Conda environment. And therefore you see that we only have a few packages here. And now we're going to, all right, let me check. So we're gonna install the Python library in the requirement.txt. So we already have it here. Okay, so no need to download it into your computer. So you want to just go and type in pip install -r requirements.txt. Okay, so this will allow you to install the contents. Let's have a look before we do that. So I'll use cat to see the contents of the requirements.txt file. And so here we're going to install Streamlit version 0.7, Pandas 1.1.3, NumPy 1.19.2, Pillow for the image, and also the scikit-learn. Okay, so we're installing only a few libraries here. So instead of typing in one by one, we just type in a single line and it will install all of the library in the requirement.txt file. So you want to type in pip install -r requirements.txt, enter. And so it will now install the Python libraries. So that should take a few moments. And the great thing of using Conda is that it will install all of the prerequisite library as well. So notice that SciPy was not included in the list, but then SciPy is a prerequisite or dependency for one of the libraries. And so it is installing that as well. So after that, we're going to install the RDKit. And RDKit is a Python library that allows you to do a wide range of cheminformatic tasks. Like, for example, you could display chemical structure images. You could read in Smiles notation. You could then convert the Smiles notation into other format like MOL format or Mo2 format or even STF. You could convert it into a three-dimensional structure. You could perform molecular mechanic optimization of the structure so that you get a relatively good 3D structure, right, with the low energy conformer. And you could also compute various types of molecular descriptor, molecular fingerprints, and so the RDKit library will allow you to do a lot of tasks. You could even search by the substructure as well. Okay, so if you have RDKit, you could convert it. You could convert molecules into numerical values, and then you could use scikit-learn to build the model. Okay. And then in this particular tutorial, we will use Streamlit to build the web application. And Pandas to create data frames of the data set. Okay, so this should take you a while. So let's see. In the meantime, I can show you the RDKit website. Okay, so this is the RDKit website. Let's have a look at the documentation. So this documentation provides you all of the essential information, like here, how you could import the chem function, and then you could use the chem function to read in molecule in the SMILES notation. Okay, or you could read it in from a mol file right here, .mol. And then whenever you read in a molecule, whether it be a SMILES notation or whether it is a molecular file, either a MOL or a STF file, it will convert that into a RDKit molecule object. Okay, so it will be this object. And then afterward, you could use the object here to compute molecular descriptors, which we will be doing today. Okay, so here, let's have a look at the contents again. Okay, so you could, let's see what you could do here. So this documentation is very comprehensive. So it is also a cookbook integrated in here. Like here, you could draw a molecule inside a Jupyter Notebook. Okay, so if you activate this, you could display the structure of the molecule in a Jupyter Notebook. And you could iterate through your entire data set, and then you could display the particular compound that you would like to see. Right? You could show the atomic properties alongside the structure. You could annotate it. You could also highlight the substructure in your molecule. You could color the atoms as well. Right here, you could highlight the substructure. Okay. So a lot of things that you could do with the RDKit. And so if you're into like bioinformatics or chem informatics, then you need to use this library. Okay. All right, so let me head back to the Conda terminal. All right, so now the libraries has been installed in pip. So let's install, one moment. Let me show you this. All right, so now we're going to install RDKit by using conda install and then RDKit will be installed from the Conda Forge channel or also RDKit channel, either one. And then the RDKit library is specified here. Okay, so this might look confusing. So let me show you something first. Okay, so let's say that I would like to install a library. I would type in conda install. And let's say that I would like to install Jupyter. I would type in like this, conda install Jupyter. Okay, but if I type in conda install RDKit, okay, it wouldn't work. Okay, it cannot find RDKit in the default repository of conda. And therefore, we have to specify the channel. Okay, so the channel name. So that is from the option here, -C. Okay, so we're gonna specify the channel name, conda install, and then the channel name. So look for it in Conda Forge and then RDKit. Let's go for that. So RDKit should be available from two channels, either Conda Forge or also RDKit. Okay, so this is an extra. So you could also delete this if you want. You could use either one, either RDKit or Conda Forge. Let me share the screen of the terminal now. Okay, so I'm not sure if you saw it. Let me type it again. I typed in conda install -C, conda -forge, and then RDKit. Okay, because -C conda forge is the name of the channel that will allow me to look for the RDKit library. Enter to install RDKit, because if you're not using Conda, you could install RDKit manually, and it's going to be quite difficult to do so manually. I can show you. Because if you do it manually, you have to install other library as well as the dependency. So that might take you a couple of hours. Here, if you want to build it from source, okay, from Conda, but let's see, installation. Yeah, so if you go to the documentation here under installation, there's many ways for you to install Conda. And so it might look quite confusing because there's so many approaches that you could do. But if you follow the approach that I'm showing you here, it will be quite simple. Just using one line, and then you could install RDKit. Let me go back. But for those of you who are using Jupyter Notebook, it might be a bit tricky to use Conda inside the Jupyter Notebook. But I've also provided a video on my YouTube channel where I show you how you could install Conda on a Jupyter Notebook. And then you could install RDKit there. So this should take a short moment. And so most of this tutorial will probably involve a lot of installation of the library here. So normally in my tutorial video, I would just fast forward this installation process. Okay, there you go. So it's installed already. And let's check it out by typing in Conda list. Let's have a look at the libraries that we have so far. All right, and so you can see that now we have a lot of libraries and dependencies installed in our Conda environment. Okay, let's try it out. Let's type in Python. Let's try import RDKit. All right, it works. So if it doesn't work, it will print out like some error message. Okay, so now it works, and note here that we have Python 3.7.9, which we have specified to be installed here. Okay, let me quit it. All right, and so now we're ready to be running the web application. But before doing that, let me show you the Jupyter Notebook. How should I do this? So we have to install Jupyter Notebook. So Conda install Jupyter. Because we're going to show you how you could run the Jupyter Notebook file, which is right here. Let me share. Which is right here. So we're going to run this in order to build the model. Okay. All right. One moment. So what time zones are all of you coming from?
Mostly from India, IST, sir. Indian Standard.
Okay. So we're at nighttime right now, right?
Yes, sir. 9 p.m.
Okay, right. So it is 10:30 for me. So it is probably 9 o'clock for you guys, right? 9 p.m. Okay. One moment.
In the meantime, we have one question, sir. Can I go ahead?
Sure.
So one person is asking, for example, I have 26 compounds. For these compounds, there is a data set in CSV format, which includes descriptors that he has calculated around 5,000 and
Okay.
IC50 values of compounds. He would like to perform some QSR analysis with the deep learning and artificial networks and predicted new IC50 values. He did not calculate any descriptors for his molecule, but he just gave numbers 1, 2, 3, 4 to the molecules.
Okay.
He's asking some program, some code, any code, sample code can do that.
Right. So he has 5,000 descriptors.
Yes, sir. Yes, sir. You can see in the chart, sir.
Oh,
Okay.
Let's see.
So,
Chat. Okay. Okay, I have 26 compound. For these compound, there is a data set in CSV format that include descriptor. Okay, so there's a CSV file that contains 5,000 descriptors, and it has the IC50 value, and he wants to perform, okay, neural network to predict the IC50. Okay, so the question is, I will not calculate any descriptor for my molecule, and I just gave numbers like 1, 2, 3, 4 to the molecule. Okay, so 1, 2, 3, 4 is probably the name of the molecule. I put the calculate descriptor for all complex in the CSV from some external program. I want to use the CSV and create my model with deep learning. Can a code sample be made about it? Or how can this be shown in existing program? Okay. Yeah, so the concept is quite the same. Whether you use deep learning, you could use Keras library, or you could use PyTorch. But the tutorial that we're using today, we're using scikit-learn. Okay, so instead of using scikit-learn, you could use the PyTorch or Keras or TensorFlow. But nowadays, Keras is already integrated in TensorFlow, right? So you could use TensorFlow or PyTorch. Instead of the scikit-learn that we're going to be using today. And you said that the question said that he already has 5,000 descriptors. So you don't have to calculate the descriptor that we're going to be calculating here. Okay, so for this one, we have the descriptors calculated in RDKit, which is mol-log P, molecular weight, rotatable bonds, aromatic proportion. So here is a rather very simple example of a QSAR model or machine learning model where we use only 4 descriptor. But in the example of the question, you already have 5,000, okay? And so the same thing, you have 5,000, so here we have 4. And you have IC50, but here we have the log S, okay? But I would recommend you to convert the IC50 value to the PIC50 value, okay? Because if you look at the distribution of your IC50, you will see that your distribution might be clustered. And so therefore it is recommended to calculate the negative logarithmic value of your IC50 in order to make it more uniformly distributed, okay? And then you could apply the same approach that we're using in this tutorial that I'm going to show you in the Jupyter Notebook to build the model. Okay, so I hope that answers the question.
yes, sir. Yes, sir. Thank you.
Thank you. And now we have already installed Jupyter. So we're going to run Jupyter now. Okay, so go ahead and type in Jupyter Notebook in the terminal. Jupyter Notebook, hit enter. And then all right, and so you're gonna see the Jupyter Notebook pop up here, okay? And so we're gonna run the solubility web app.ipynb, which is the IPython notebook. It stands for IPython notebook. And then later it was renamed to Jupyter Notebook. All right, there you go. So we're simply running it sequentially, okay? So here we're gonna import pandas. So what you could do is you could click on the cell and then you could click on the run. And when you click on run, it will run this. And you're gonna notice here that you see the asterisk symbol. which means that it is running. So it is a bit slow. Wait 1 more second. All right, it works now. And now we're gonna run this particular cell again. So instead of run, you could also use the shortcut. On the keyboard, you wanna press the Shift and Enter. And then you're gonna get the same thing, okay? So I like to use shortcuts. So Shift, you type Shift, Enter at the same time. Shift, Enter. Okay, and then we're gonna see that the third cell has already ran. Okay, so you can just Shift, Enter, and it will sequentially run each cell from top to bottom. Okay, so now let me explain. So here, the great thing of a Jupyter Notebook is it allows you to put your code in a documented fashion. Okay, so the two essential components of a Jupyter Notebook is here. You have the text cell. This is also a text cell. So a text cell, if you double click on it, it will become a Markdown language. Okay, so in a Markdown, you have the hashtag to represent the H1 heading. And then you have the double asterisk before and after in order to make your text bold. If you have a single one, it will be italic. Let me show you here. In italic, if you have one, right, it will be italic. So you have to Shift Enter it. And then notice here that this one is in italic form. And this one is bold, right? Because we have double asterisk. And then we also have the H tag. And let's double click on this one. For this one, we only have the H tag. If we add double asterisk, it will be bold, but it looks quite the same. We could just take it out. Okay, so you could Google for Markdown cheat sheet, and then you will see all of the commands that you could use, right? Like for example, if you click the plus, you will get a code cell. And when you get the code cell, you want to click on here, the drop down here. You could change it into a Markdown. And now you could put in text. Okay, this is like H1, and then you have 2, it becomes H2, H3. And so what is H1, H2, H3? It's kind of like this: section, subsection, subsection. Okay, and so whenever you have H1, H2, H3, so the section will be smaller and smaller. But then there are also cool stuff that you will see when you are using this in a Google Colab. If you use it in a Google Colab, you could collapse the entire H1 level. Okay, on the Colab, you could just click on the button here, and it will collapse the entire content here. And if you click on the button here, it will collapse everything underneath this particular section. Okay, so it seems to work only in the Google Colab. So that's a cool feature to have. All right, and so this one in the second cell, we're going to be reading in the data set here, which is provided on the GitHub repo. So it's the Delani solubility descriptor. So this one is pre-computed. Okay, so actually we're not using RDKits. Okay, so it is already pre-computed. And so we could copy this link and let's have a look. Let me share this. Okay, so these are pre-computed descriptors. So this is in CSV format, okay? So the value is separated by the commas as a column separator. And so the first line, you're going to see the name of the descriptors, right? We have the first value to be mol-log P, molecular weight, number of rotatable bonds, aromatic proportion, and the log S value. Okay, so actually I've shown you, so I'm not sure. Let's see, right now it's 2241. Okay. Okay. So this is pre-computed, right? So we have previously used the RD kits. So let me find the YouTube video right now. Data professor. And let's see. It's in the playlist of Bioinformatics. One moment. Let me share it here. It's in this playlist. And let me find it. Okay, it's right here. Part 1, part 2. Okay, so there's 2 parts. So let me copy the links for you. So in part 1 here, I've shown you step by step. So you can see that it takes about 25 minutes for this one. But I'm afraid that this will be too long for this tutorial. So I'm going to provide you these 2 links, part 1 and part 2, in the Notion webpage that I've shown you already. Okay, so in this video, I've explained to you what each of the descriptor mean, and then explain to you how you could compute the descriptor. Let's see. Okay. Oh, advertisement. Yeah, so this one also provides the code as well. If you click here, code, it will bring you to the code page. So you could run this particular notebook, right? So this particular notebook will allow you to install RD kits on Google Colab. So you could run this on the Google Colab as well. And then this one is about installing Conda. All right, and here we can see the data set. The data set is comprised of the name of the compound in the first column, and then it provides the value of the measured, the experimental value of the solubility. And then in the last column, it provides the SMILES notation. Okay, so we use the SMILES notation here, which is a chemical structure information. And then it's right here. And then we read it in. We read it in using RD kits, using the chem function. So in the chem function, we use the mol from SMILE. And then we put in the SMILES notation here. Right, and it will become an object, a molecule object. Right, so each molecule will be read in, and it will be a molecule object, and then we will compute the descriptor for each, shown here. Okay, so each molecule, this is molecule 1, molecule 2, molecule 3, 4, 5. And so this data set has a couple hundred molecules, 1,144 molecules. Okay, and then we iterate through the entire molecule list, right? So we iterate through using the for loop. Okay. And then we've created custom function. Right here, we define our own custom function to calculate the mol-log P value, the molecular weight value, the number of rotatable bonds, right? And then we created a data frame in pandas here. And then after we use this custom function, which takes in as an input argument, the SMILES notation. So here we use the custom function that we have generated, and then using as input argument, the SMILES notation. And then it will spit out the data frame of the computed molecular descriptors. So actually, aside from the 3 descriptors that you see here, you could modify right here, right? You could add additional descriptors right? So you could copy this line, copy it, and then you could add additional lines here, right? And then you could cherry pick the descriptors that you would like to be calculated in this code, right? You could have like additional, maybe 10 or 20 or 100 additional lines here, so that it will calculate additional descriptors. Okay, so actually that's the content for another tutorial. Maybe I'll create a tutorial video on the YouTube channel as well. So in RDKit, there's so many descriptors. Okay. Let me show you then. I hope time will allow. Okay, so I try to finish this part in 15 minutes, and then I'm going to show you the actual web app. Okay, so we will have about half an hour for that. Okay, so let me go back to the RDKit documentation. Let's find the example. One moment. Descriptor right here. Okay, so you could just find, right? I type in Command F or Control F, and then I type in descriptor, and then here, descriptor calculation. So aside from the molocp as shown in the tutorial that we're using today, you could also calculate other descriptor like the TPSA as well. Okay, so in order to look at the full options, you want to click on the link here, like the API, or actually right here, list of available descriptor. All right, so we're here. So there's so many descriptors, right? Look at this column, list of available descriptor. There's so many. Gas cycle charge, okay? And we use only a few, but then some of them might be a bit redundant. And you also have to compare, okay, which one are redundant. Number of amide bonds, the ring count, right? TPSA. Okay, and then they also have 3D descriptor as well. So you probably will have to generate a 3D confirmation of your molecule. And so that will require optimization using molecular mechanic force field as well. And if you want to have more accuracy, you could also use quantum mechanic or computational chemistry in order to optimize your molecule's geometry. And then you could calculate other quantum mechanic descriptor like the energy of the molecular orbital. And also here, you could calculate molecular fingerprints as well. And they have a couple here. Okay, and each fingerprint type here will have several hundreds, okay? Okay, so this is very nice. As you can see, there's a lot of descriptor that you could calculate. And you could feel free to add it into the custom function that I've shown you. Right here, you could add it into this custom function, right? All right, and now that we already have generated the descriptors, and then it's gonna read in the data frame of the calculated descriptor, and it will perform the data split, which I will be showing you in this notebook. So let me close this, okay? So part 1 and part 2, I'll provide you the link. Okay, let me do it right now. The link, share, copy. Let me add it to Notion. So let me share it to you. Okay, so Notion is a very good application. I'm not sure if some of you have used Notion. It's a good note-taking application, and it's very powerful as well. And it has a lot of templates. So I'm gonna make some video of this soon. So let's see, learn more, helpful. Okay, let me add a new entry, and it's gonna be, what should I call it? I'll call it Chem Informatics Part 1. And the links here. And Then I'll call it Chem Informatics Part 2. Open, and let me find the link. And part 2 will essentially be using PyCaret to build the model. And so PyCaret is a AutoML library in Python. So Moez Ali has developed this awesome library. And so you could just, in only a few lines of code, you could build machine learning models using a lot of machine learning algorithms in one run. Okay, there you go. So Chem Informatics Part 1 and Part 2 is right here. And actually, I could provide you the GitHub link as well for your convenience if you want to try this out in your spare time. Let's see, Part 1, Part 2. Okay. Part 1 is this one. Part 1. Part 2, we have 2 files. This one. Okay, so the links are provided here, the links to the GitHub repo. And let us now start create the web app. But first we have to head back to the Jupyter Notebook. So many windows open. One moment. Okay, let me share the screen. All right, okay, so we have loaded up the data frame here. And now we're going to drop. So for this data frame that we have loaded up, okay, it's called data set. Okay, I could create a new cell here. I could type data set, shift enter, and then it stays data frame. So what I want to do now is I want to split this into X and Y variable, okay? So the X variable will contain the first 4 columns, and then the Y variable will contain only the log S column. Okay, so I'm going to split log S to be variable Y, and then the first 4 variable will be the variable X. Okay, and therefore I created here X equal to dataset dot drop, and I'm going to drop the last column, the log S column. Okay, and then I specify it to be X variable. And therefore you see that the log S column is now gone. Okay, and now we're going to specify the last column here, the last column, -1, the last column to be the Y variable. Okay, so this is one way of doing it. Another way would be to say Y equals to dataset log S. Let's see the name. Log S. This, okay. Same thing. You get the same output. So there's many ways. Or you could also do it like this. Y equals dataset dot log S. Let me show you. Okay. So there's 3 ways of doing the same thing. Okay. So select whichever approach you like. Okay. And so we get the Y variable to be the log S column. Okay. And now let's have a look. Let's have a look at the X variable. And let's have a look at the Y variable. Okay, now we have X and Y. So we're good to go. Now we're ready to use scikit-learn to use X and Y to build the model here. Okay, so in this tutorial, we're not going to perform any data split. We're just going to use XY, the full data set, to build the model. But then in your own custom workflow for your own research project, I would recommend you to perform data splitting as well. And so here we're gonna use a basic linear model. Okay, so we're gonna use the linear model function from the sklearn. And then in order to evaluate the model performance of a regression model, because here we're gonna build a regression because the y variable is quantitative. Okay, so it's a floating number. Okay. So, however, if we want to do classification, then we're going to use different metrics here. Okay. So for regression, we're going to use the R² value and the mean squared error, or we could also use mean absolute error as well. Okay. So up to you. But then for this tutorial, we're just going to use mean squared error. Okay. So run the cell. And then we're going to create a variable called model, and then we're going to assign the linear model.linear regression function to this variable. And then we're going to train the model by using model.fit. And then the input argument will be x and y. And x and y has been created here as we have done previously. Okay. So it's taking quite some time to load it on my local computer. And let's train the model. Shift enter here. Okay, and the model has been trained fairly rapidly. And let's make the prediction. Okay, so model prediction. So as you can see here, the Jupyter Notebook is documented so that you could read it. Okay, you could also provide more explanatory text below here. Like for example, this is here, or we will now perform the prediction by applying the trained model. This will be performed by using the model.predict function. And then you could use that. You could highlight this by using the tick. Right, and you notice that it has this like gray color on the background as well to highlight it. Okay. And then the text font looks a bit different. So this would highlight the function name. Okay, so you could, if you use this, let me show you. Okay, so you could use like superscript by using the $sign before and after. And then this would represent the superscript. If you want to make it like, let's see, for evaluating, like for example, if you have the term IC50, you want to do it like this. Okay. And note that you need to have the opening and closing braces on the 50, because if you do it like this, let me show you. Then only the 5 will be in the subscripts. Okay. Therefore, you need to have the braces around the 50. All right. So this is just to show, but it doesn't make sense because in this example, we're not using the IC50. Okay. So we use the R² and the MSE. Okay. So MSE is the mean squared error. Okay. All right. And so we're going to apply the model.predict function, and then we're going to use the X variable. And then we're going to assign the prediction into the y_pred variable. And then the predicted value is shown here. Okay. And if you want to convert this into like a data frame type, we could use it into a series. And then we could say ypred. But then we're going to assign this to, let's create a new variable. Let's call it ypred without the underscore. And then, oh, it's not defined. Okay. Because this is capital, right? So we have it here. And so we have this as a column now, because before it was as an array, okay? So here we're using the pandas series. And let's see, what if I want to combine it to this data frame again? This is X, right? But yeah, okay. So I have the X data frame here, right? I want to combine these two data frames, X and ypred, okay? So let me show you how you could do that. You could do pd.concat, and then you want to use the opening and closing bracket, and then you just type in X, the element that you want to combine, and we're going to combine ypred. So just type in ypred. And now you want to do X is equal to 1, shift enter. And now you see the predicted value is shown here. Okay, but let's say that we want to show it alongside the actual value Y. So we could also do, we put the Y first, and then we put the ypred next. Okay. Notice that this one doesn't have a variable. Is it name ypred? Or let's call it log s pred. Okay, so I added the name to log s pred to the ypred variable, and let's combine it again. And there you go. The name of the column is updated, and then I have to assign this into a data frame in order to save it, right? And then now I have it as a data frame, right? And let's say that I want to save this into a file, then you could just do df.to.csv, and then you could name the file. You could say results.csv, hit enter, and then you're going to get the CSV file. Let me find it. Okay. So it's right here. So this is the resulting file. Okay, so we're right here. Log s pred is right here. Log s is right here. Okay, so you see the actual value, and then you see the predicted value here. Okay, and so you could save this as a file. All right, let's continue back to the Jupyter Notebook to actually build the file, and so that we could actually build the web application. Okay, so we have 25 minutes left. Let's go back to Jupyter Notebook. All right, here. So I just added this as a bonus in this tutorial, just in case you're wondering. Right now, we're gonna print out the model performance right here. So you can see the coefficient of the regression coefficient. Okay, so, you know, like in a typical, let me add more explanation. Like in a typical linear equation, you have like this. For this one, we have log s equals to the 4 parameters, right? We have mol log p. We have mol weight. we have number of rotatable bonds and an aromatic proportion. Okay, so here we have the intercept to be this value. So that's our equation. And then we have AR to be here. It's right here. It's sequential. So we have this one multiplied by, and then this one is right here. So this is our equation. And then this value is right here. This plus, right? This minus this one, minus more weight. And then the first value is right here. There you go. So that's our linear regression equation. So this is the equation generated by this particular model. Okay, and to get the intercept, it's right here. You use this function, model.intercept. To get the coefficient, you use the model.coef. Okay, to get the mean squared error, it's the mean squared error function. And to get the R2 score, you use the R2 score function. And the input argument will be the actual value, y, and the predicted value, y pred. Okay. And so you have the R square value to be 0.77. And then we have the mean squared error to be 1.01. Okay. And then, oh, actually, I created this already. Okay. Right. So in reverse order, I have this one right here. Okay, so I've already shown you how to do it programmatically already. So this equation is generated automatically. Okay, so even if you add additional data to this data set, it's going to generate this equation automatically for you. Okay, and now let's create the visualization of the predicted value. So this is the plot that you have generated, right? So this is the experimental and the predicted log S value. So it looks pretty good. And now we're going to save it out. Okay, we're going to use pickle function here to dump it, to save it into a pkl file. Okay, and so let's do this. So let me make a note here is that if you want to save the model out, make sure that the model that you saved here is done locally. Because if you do it on the Google Colab, the thing is Google Colab might be using a different version of scikit-learn, and therefore it might not be compatible with your web application. Okay. So you might be using a different NumPy version. You might be using a different scikit-learn version. And so the pkl file that is generated will not be the same. Okay, so you want to do it locally on the computer that you want to run the web application. Okay. So let me show you the contents of the web application. Take a look at the solvability app.py. Okay, share the screen. Okay, right here. Share. Adam, okay. Okay, this one better. All right, so here. So let's have a look generally. So this particular web application is only 110 lines of code. Okay, so that's not a lot of lines of code to make this fully functional web application that you have saw. And so the first couple of lines here, we're going to import the libraries, right? We're going to import all of the necessary libraries that we're using. So you can see here that we have Pickle, which is the trained model that we have done already. Because the thing is, we don't want to retrain the model over and over and over again, right? Because the data set is the same. So we just want to train the model one time And then we deploy that trained model. And then the web application will take the query molecule, the input molecule, and then it will use the RDKit to perform the molecular descriptor calculation. Okay. So just a moment ago, I told you about how we created this custom function to calculate the molecular descriptor. So we have to put the custom function inside the code here. Okay, so we documented that here, custom function. So lines number 15 until 57 is the custom function to calculate the molecular descriptor. And then lines number 63. So this is essentially just gonna display the logo of the web app. Let me go back. Let me find the application. Oh no, okay. We have to run the web app now. Okay. All right, so let me close the Jupyter Notebook. So let's go back to the terminal, and I want to Control-C to exit that. And then it's asking me whether I want to shut down the notebook server. I say yes. Oh, it's resuming because I took longer than 5 seconds. So let me do it again. All right, now I exited that. And now I'm going to run this solubility app.py. Streamlit, run solubility app.py. All right, and now this is the web app. Okay, so the image variable that I have talked about just a moment ago is this image that I have drawn, the logo of the web application. Okay, so let's go back to the code. Right, and then we use the st.write function in order to display the heading of the web application. As I mentioned already, we use the h1 tag here, which is the hashtag. So the font will be big. So it's a heading tag. And then we describe the web application here. And then the side panel will be shown here. Let me show you back to the web application. So note that on the side panel right here, on the left-hand side, you have the text box that takes in the SMILES notation as the input file. I mean, as the input text. So each will be a molecule, right? So for example, I could modify it. And then after that, I want to press Shift. I want to type in Command Enter to apply it. And now the prediction, as you see, is right here. It's updated. So this thing here, the third molecule notation matches this third notation here. Okay. So this is the side panel. And let's go back to the code. It's right here. Where were we? Right here, SMILES input. Right here, st.sidebar.text area. So text area function will allow us to have the text box, and st.sidebar will tell us the location that we want to place the text box. So if we delete this part here, it will not be placed in the sidebar, but it will be placed in the main panel. Okay. So we're gonna leave it there. Okay, so here we're just taking in the SMILES notation, and then this is the default input that you saw, the 3 molecule that you saw. /n is the new line. It means to hit on enter. Okay, so there will be enter at the end of the first line, and then another enter at the end of the second line. All right, and now after that, it's going to calculate the molecular descriptor. and then it's gonna here assign the calculated descriptor to the X variable. And then essentially that's all, right? On lines number 103, we're going to load in the trained model. Remember the trained model that we have used in the Jupyter Notebook? It's right here, solubility model.pkl. And now we're going to load in the model, and then we assign it to the load model variable. And then we're going to use load model.predict. And then we're going to use the X variable here. X is right here, the molecular descriptor that we had generated. And so it will use the molecular descriptor that has just been calculated from the user input from the side panel. And it makes the prediction, and it assigns the prediction into the prediction variable. And then it will place the prediction underneath the st.header here. Okay, and that's it. That's essentially the web app. Let me go back and show you the web app. Okay, so I've told you that it will display the computed molecular descriptor, and then it will display the computed log S value, which has been predicted. Okay, and so as you can see here that this is a very simple web application. And so I have already explained to you that we initially loaded the necessary libraries for this one, like Streamlit, NumPy, scikit-learn, RDKit, right? And so the user will put in the molecule that they want, right? And then command enter or control enter, and then the app will run the prediction in real time, okay? In real time, okay? And it makes the prediction. And so it takes in the input molecule as a SMILES notation. It performs the molecular descriptor calculation, right? It displays the input molecule that you have placed in the text box. It displays your input here. It then computes the molecular descriptor, and it shows the computed molecular descriptor here. It takes these computed molecular descriptor and apply the model.predict function to make a prediction. And then the resulting prediction value will be displayed here. Okay, and so this is essentially the web application contained in the app.py file. Okay, and I think we've done it. Okay, so we have 10 minutes left for questions and answers. So I'll be happy to entertain any questions that you may have.
So, I have a question. With this model, we can predict the descriptors for a new molecule?
Yes. So we're not predicting the descriptor, but we could calculate the descriptor, and then we could predict the solubility value.
Okay.
Right. So the RDKit will be used for calculating the molecular descriptor.
Yes, Afrin Khan. You can unmute.
Hello, sir. Thank you for a wonderful explanation. Sir, I wanted to know for any QSAR model, they ask for applicability domain. So how could we calculate that in machine learning?
Okay. So actually, we've done a couple of research articles where we calculate the applicability domain. So normally we like to use the principal component analysis. So you could calculate the applicability domain by performing PCA analysis on your data set. And then you could also do the same for your training set and then your test set. So if you perform data split, right, you could do the PCA of your training set, of your test set, or of your other external sets. And then you could visualize the distribution of the molecule in the scores plot And so that will essentially be your applicability domain. So if the new compound or the test compound fall outside the applicability domain, meaning that they do not overlap, it means that a new compound is not predictable. So it is outside the applicability domain. And so actually I've never done a web app for that. Maybe that could be a good idea for a future video as well. So yeah, I'll probably make a video showing you how to do that.
Thank you, sir.
Rohit.
Okay, sir, I have one question actually. That's
Right.
a nice presentation.
Thank
So
you.
as you have told that we can save pickle file if from any scikit-learn version, and after
Right.
that where we want to use in different Python version or scikit-learn version, so we cannot use. So like I have saved one pickle file from suppose scikit-learn 1.9 or something, and if someone is downloading, like I have given my code to the GitHub, so if someone is downloading my code, so he can use or he cannot use, or any other
Right.
method to save the pickle file so anyone can use in any scikit-learn version.
Right, that's a great question because on one occasion where I trained the model on the Google Colab, and then I downloaded the pickle file into my local computer, and then when I try to show the web application, I try to run it, and it gives me an error that, okay, you're using a different version of scikit-learn. But then when I trained it locally and then I used the pickled file, so there were no issues there. So there might be some compatibility issues. So I would recommend you to share the code, and then the user can download that code, and they could also train it locally on their own computer.
Okay, thank you, sir.
Yes, Jyotirmay, do you have some specific question? You can ask. Okay. Yes.
Hello.
Yes, Harikishan,
Hello.
go ahead.
Suppose I want to do this training without the descriptors. I just want to put the smiles into the training model, like a regression model or something. How can I do that? And which model would be good for such case?
You mean you want to use the smiles, but you don't want to calculate the descriptors?
No, without using descriptors. Suppose if I means I just want to take the smiles, read it and put it in the training model. Is it possible?
It is possible, and actually we're doing that as well. So for that, the thing is you're bypassing the descriptor calculation, and so therefore the descriptor will be the smile notation. And so right now we're experimenting with using that. We're experimenting with using the smiles notation, and then we're performing tokenization, meaning that we're going to split the smiles notation into the fragments. So each character will be kind of like a descriptor in itself. Like for example, in a typical smiles notation, here, let me show you. So in the side panel here, you see that NCCC is a molecule, right? So it will tokenize it, meaning it will split it into NCCC, and then it will essentially see it as like a character, right? And then it will compare. So if another molecule contains N in the first position, and another one contains N in the first position, it will be the same, right? And it will have, like for example, you could represent it as an image. And so this will be like maybe have a value of 1. So the thing is, does it contain N in the first position? If it does, then it will have a value of 1. If it have a value of 1. If it doesn't, it will have a value of 0. So therefore, you will perform tokenization on your smiles notation, and then that will be the descriptor. And then we'll have to use, and then we could use like deep neural network, like LSTM, or you could also try out the scikit-learn as well, using random forest. So right now we're experimenting with that, and hopefully we'll publish a paper and maybe make a YouTube video about it.
Thank you, sir. Rohit, can you conclude, Rohit?
Okay, thank you, Ibrahim, and thank you, Professor Chenin. This this is an excellent lecture and everything was crystal clear. And I hope all the participants understand everything. If they want to enhance their knowledge, so they should visit the Data Professor YouTube channel and they can see the video and they can ask the question from Professor Chenin also. He has summarized and he has also showed by hands on that how you can develop a regression model, and this is very good for the advert prediction. When you like you are using some tools in the web designing for the advert prediction, but most of the user don't know and they don't read the paper that how our model or how our computer or we are giving the input as like compound, and it is calculating the descriptor, and on the basis of descriptor it is giving the values. So generally the user don't think, so maybe they can correlate with that regression model. And one more thing, that they can also use that regression model in their research purpose when they want to develop any QSAR model. So I hope that everything was crystal clear, and thank you, Professor Chenin, for accepting the invitation.
Thank you. It's my big pleasure. And actually, let me make a note here that if you're interested, you could also go to my academic GitHub as well. Let me show you on the screen. So let me find that. Is it right here? Yeah. So my academic GitHub is github.com/ChannonLab. And so these will have repository of research articles that we also published. So for example, in the HCV Pred here, if you search for this article, we have already published it. We published it in the Journal of Computational Chemistry. And so you could read the full article here. And then the full text, the full code is provided here. So we provide you with the curated data set. We provided you with the code and the descriptor, the R code that you could use to reproduce our work. So here we use R, and then you could perform our model building from the code. And we also shared the code to several of our other papers as well. So you could check out the GitHub here. This is the first GitHub. And why don't I put this in the chat? Let me add it to the chat. One moment. And another one would be Channon N. So another one would be my GitHub for the Data Professor YouTube. So all of these three, so the GitHub of Data Professor will contain the files and the code used in the YouTube tutorial. Channon N and Channon Lab will contain the code that we use for our research publication. Okay. Yeah,
Thank
like
you,
for
sir,
example
for
here.
the information.
Right, like if you
And
click here,
you are gently contributing towards the science, because you are creating the code and you are also creating the Jupyter Notebook. And I think that GitHub is the most useful thing to learn the data science and other things in the machine learning. Because okay, thank you so much, sir.
Exactly. Thank you so much.
The session is over to Ibrahim.
Yes, sir. Yes, sir. Kalyani ma'am, can you thank Channon, Dr. Channon? Dr. Kalyani is also one of the organizing secretaries.
Okay.
He's with us. I think she's having some mic problem. I am going to end the session, sir. Thank you so much for accepting our kind invitation, Dr. Channon, sir. We are looking forward to work with you, sir. More collaborate with you. Thank you so much, Dr. Channon, sir. I'm going to end this meeting now.
Okay. Thank you so much, everyone, for attending.
Thank you, sir.
Thank you.